So this is chapter 12 on the cell cycle. The cell cycle is the life cycle of a cell, and it's looking at the time from when the cell is first formed until it divides in two. So with cell division, it's going to result in two genetically identical daughter cells. So you've got your cellular genetic material, where the genome is going to be considered the cell's genetic information. It's a pretty generic term used for the genetic information. It does not really specify what form it's in. The chromosomes are the packaged form of the DNA. They're going to be in this form during cell replication and distribution. When we look at cell division, it's going to make a difference, particularly in animal cells, whether or not we're looking at a somatic cell, which is all of the body cells except the reproductive cells. It's everything except the sperm and egg. Those cells will reproduce differently than your gametes, which are your reproductive cells or your sperm and egg. The sperm and egg are going to have half as many chromosomes as the somatic cells because those are going to join with somebody else's reproductive cells in there, and the new cell will have DNA based on what it inherits from the two parents. So chromatin is the complex of DNA and the associated proteins. It's the loosely coiled DNA, and this is the form that it will be in most of the time. So during eukaryotic cell division, the DNA is going to start out initially as chromatin fibers when it's not dividing. And even as it starts to duplicate the DNA, it will be in chromatin. After duplication, it's going to condense into the chromosomes. The sister chromatids are the two parts of the chromosomes, and they're going to each contain identical DNA molecules. When you picture the X shape of a chromosome, each half of the X is one of the sister chromatids. The cohesins are the adhesive protein complex that's going to attach the two sister chromatids together. So your sister chromatid cohesin is going to be what is attaching them in there. The centromere is the specialized region of the chromosome where the chromatids are most closely attached in there. So the cohesin is the protein that's going to hold them together, and the centromere is the region of the chromosome itself where it's held together by the cohesins in there. So later on in cell division, you're going to have the two sister chromatids separate, and they are each considered individual chromosomes. Each nucleus of the new cells will receive a collection of chromosomes that's identical to that of the parent cell. So when we look at the picture here, you've got your centromere in there and your centrosome, or your chromosome arm in there, which is part of the sister chromatid. You've got your two sister chromatids here, so each of them is one leg of the X. So in cell division, you're going to have the chromosome duplicate first, which will include the DNA replication and it condensing into the chromosomes. And then when it divides, you're going to divide and have one of those sister chromatids go with each of the two new cells. So mitosis is considered the process of the division of the nucleus, and cytokinesis is the division of the cytoplasm. Usually you're going to have cytokinesis follow division of the nucleus in there. In human cells or animal cells, generally cytokinesis is going to occur at the same time. There are some cells that will have just division of the cytoplasm and not necessarily cytokinesis that will follow it. With meiosis, this is your sex cell division that will give rise to non-identical daughter cells with only half of the chromosomes as the parent cell. This is what your sperm and egg will have. So those cells will have half of the genetic information from each parent, so when it fuses and forms the zygote, it can have half the genetic information from each parent. So when fertilization occurs together, that's when you fuse those two gametes together, and then the number of chromosomes will return back to the normal number. So we can look at the different phases in cell division here. You've got the mitotic phase here, which is just a short division or a short portion of the circle. And then you've got interphase here, which is the majority of the circle. Interphase is the non-dividing phase of a cell's life cycle, and the mitotic phase is the dividing portion in there. So the interphase is also divided up into different phases as well. We can refer to the mitotic phase as M phase, it's the shortest. Interphase is 90% of the cell cycle. This is when the cell is going to grow, it's going to make copies of its chromosomes, it's going to prepare for cell division, as well as doing its normal cell life functions. G1 phase here is the first gap, and this is primarily cell growth. During the S phase here, they call it S for DNA synthesis in here, your chromosomes are going to be duplicated and the cell will grow some more. Then you have G2 for the second gap phase where it grows even more and it completes preparation to go into the M phase here. And then mitosis can be broken down into 4 or 5 phases depending on the source that you are looking at in there. The phases are prophase, prometaphase, metaphase, anaphase, and telophase. And it looks like telophase has disappeared out of your notes, so you will want to make sure to add that into your own notes yourself. When it's broken down into 4 phases, if you look at one of those textbook sources, they just look at prophase, metaphase, anaphase, and telophase. Your particular textbook uses 5, so I will stay consistent with that in this class. So your mitotic spindle begins to form in the cytoplasm during prophase. And here you can see part of the mitotic spindle forming across here. It's made up of the microtubules and proteins, and it will elongate by incorporating more tubulin subunits on there, and then it shortens by losing those subunits. So the tubulin gets longer. The centrosome is going to organize the cell's microtubules and start the assembly of the spindle. And then the centriole, or pair of centrioles, is going to be located at the center of the centrosome. We know that it's not essential for cell division, and it is not present in plant cells. During interphase, a single centrosome replicates to make 2 centrosomes that remain near the nucleus and then move apart in prophase and prometaphase so that they end up at opposite ends of the cell. So here you can see where the centrosomes have moved to the 2 opposite ends of the cell here. The aster is a radial array of short microtubules that are going to extend out from the centrosome. So right here is where you have the aster being formed. You've got the centrosome and then you've got these microtubules coming out from it. The kinetochore is the proteins associated with specific sections of the chromosome DNA at the centrosome. So where you have your centrosome and your DNA in there, that region of the DNA is your kinetochore in there. It faces in opposite directions, and during prometaphase, some of the spindle microtubules will attach to the kinetochores. When the kinetochore is captured by the microtubules, that's going to help to pull the chromosomes and move them towards the poles. Your metaphase plate is going to be here in the middle, and you can kind of see where they've created an imaginary plate in here. And that's going to be between the 2 spindle poles, and that's where your duplicated chromosomes are going to be located during metaphase. So here you've got your duplicated chromosomes on there, you've got your kinetochores in there, the kinetochores microtubules sticking out from it, and then you've got the overlapping that you can see there. During anaphase, this is going to be when the cohesins that are holding the sister chromatids of each chromosome is going to be cleaved, and it's going to be an enzyme that will cleave that so that they become separate chromosomes. So your non-kinetochore microtubules are going to be responsible for elongating the whole cell in anaphase. Your non-kinetochore microtubules are going to be from the opposite poles of the cells and will overlap. They're pretty extensive during the metaphase portion, and the region of overlap is reduced in anaphase as the motor proteins walk away from each other. So we'll go into more detail on that a little bit later on. In telophase, you're going to have a nuclei reform. Again, cytokinesis generally is going to be beginning. Depending on what type of cell it is, it may be closer to wrapping up and beginning more in anaphase or in telophase. So during G2 interphase, your nuclear envelope is going to be bound to the nucleus or one or more nuclei. Your centrosomes will form, and the chromosomes are going to form during the S phase. So they have not yet condensed. So here you've got G2 of interphase here where your chromosomes have not yet condensed in there. You've got your centrosomes with the centriole pairs. Your chromatin is being duplicated. You still have a nucleolus, a nuclear envelope, and the plasma membrane. When you go into prophase here, you're going to start that early mitotic spindle in there. The chromosomes are going to consist of the two sister chromatids in there. You've got your centromere, so they have formed in there. In prometaphase, you've got fragments of the nuclear envelope is about all you have left. It's been broken down. You've got the kinetochore microtubules here, and then you've got your non-kinetochore microtubules that are forming in there, and then you've got the kinetochore you can see on the DNA. During metaphase, you've got the metaphase plate. What is going to be your classical thing that happens here is you're going to have the chromosomes align along there. And then the centrosomes are at one spindle of the pole and the other spindle. During anaphase, your two chromosomes are going to separate into daughter chromosomes in there. And then during telophase, you've got the cleavage furrow. Sometimes you may see the cleavage furrow starting to form an anaphase in animal cells in there. You're going to have a new nuclear envelope form, and you're going to form new nucleoli in there. You've got your two new daughter cells that will occur. So underneath here in your notes, we've just got written out where those different steps will occur. Metaphase is the longest of those phases in mitosis. So when you have your kinetochore here, and you've got your microtubule, you've got this motor protein that's going to help to move things in there, to help move the chromosomes when they separate in anaphase in there. So what's going to help move it along there is you've got these tubulin subunits that can be added on and broken down to move the chromosome along the microtubule in there. So cytokinesis is the division of the cytoplasm that's usually well underway by late in telophase. It may even be close to finished up in there. It's going to involve formation of a cleavage furrow in animal cells where it's going to begin to pinch in two. So here in an animal cell, you can see where this looks like kind of a crack or a fold here. That's your cleavage furrow being formed in there. It's a shallow groove in the cell surface near the old metaphase plate. The cytoplasmic side of the furrow is going to have a contractile ring in there that's got actin filaments that are going to interact with myosin, and that ring is going to contract like a drawstring on the inside until it's pinched in two. In plant cells, instead, you're going to have a cell plate in there. There's no cleavage furrow because the cell wall is going to be harder and tougher. The vesicles from the Golgi are going to move along the microtubules to the center and then coalesce to produce a plate in there that will divide the two cells. So in here you can see where that contractile ring is on the inside. Here you can see where those vesicles are forming the plate to separate the two cells. Another type of cell division we have is binary fission. This is asexual reproduction that will occur in some of the single-celled eukaryotes. It also is quite common in prokaryotes. Both your bacteria and the archaea will do binary fission. but they don't actually do any mitosis, because instead of having the X-shaped chromosomes like you see in the animal cells, your human cells, and many other cells, instead they're going to have a single circular DNA. So you don't actually have chromosomes to separate in there. So in here you can see you've got an origin of replication in there in the bacteria, cell wall on the outside, the plasma membrane in there. And what will happen is you've got this origin of replication where you're going to begin to make 2 copies of that. And then you end up with those 2 copies separating in there. And you end up with 2 daughter cells. So at the origin of replication, this process of cell division begins at a specific place on a circular chromosome. When you start to separate that chromosome in there, it's going to produce 2 origins, because you can have it proceed in 2 different directions away from the center where it's separated. When the chromosome replicates, the cell is going to elongate to about twice its size. As the plasma membrane grows inward, it will begin to divide it into 2 daughter cells. A lot of times the DNA is going to be partly attached to the plasma membrane, and that's going to help to pull it, separating it into the 2 cells. So when we look, where did mitosis come from evolutionary, in terms of looking at evolutionary terms of it? Mitosis, they figure, had its origins in the prokaryotic methods of binary fission. And some of the proteins involved in prokaryotic binary fission are related to eukaryotic mitosis proteins. So that's why we figure that that was the origin of it in here. There is some possible intermediate stages in evolution that we can see in the way some other organisms will divide, such as the dinoflagellates and the diatoms or yeasts. So your dinoflagellates are going to be replicated with their chromosomes attached to the nuclear envelope, and they will separate as the nucleus elongates prior to dividing. So here you could see that you've got it attached to the nuclear envelope in there. The diatoms and yeast, they have a spindle within the nucleus that's going to separate the chromosomes. So you can see the spindle here separating the 2. And then when you compare it to how it's done with most eukaryotic cells in there. So your eukaryotic cell cycle and molecular control of the cell division is going to be a little bit more complex process than what you may originally have imagined. The timing and the rate of cell division is going to vary with different types of cells, but it's not random. It's going to be dependent on what is going on around the cell. So how do we know this? Your evidence for the cytoplasmic signals, your cell cycle is going to be driven by specific signaling molecules in the cytoplasm. And the way they figured this out is they tested it by fusing cells that were in different phases of the cell cycle. And so if you had a cell that was in a little bit later phase of the cell cycle, it actually helped to push it along into cell division in there, so that you ended up with it moving on to the next phase in there. So when a cell was in the S phase and it was fused with a G1, a cell that was in the G1 phase in the nucleus, the G1 nucleus immediately enters into the S phase in there and catches up with the S phase in there. So it's moving into what is going on with the signals that are in the cytoplasm. When the cell is in the M phase and fused with a cell in the G1 phase, the G1 nucleus immediately begins to go into mitosis to catch up with things. So these things were evidence that there is messages in the cytoplasm that tells the cell when it is time to divide. So we know we have a control system in the cytoplasm, and it operates cyclically with a set of molecules in there to trigger and coordinate events in the cell cycle. Another interesting thing we have about this is we have checkpoints in there, control points in the cell cycle where there are stop and go signals that are going to regulate things. In animal cells, they generally are going to have built-in stop signals that have to be overridden before you can move on to the next phase in there. A lot of the checkpoints are going to come from surveillance mechanisms in the cell. So your G1 checkpoints, sometimes referred to as the restriction point in mammals, if a cell gets a go-ahead at G1, it's going to go ahead and progress through the G1 phase, the S phase, the G2, and the M phase. If not, it's going to exit and go into the GO phase. The GO phase is a non-dividing state of cells. Most of the human body cells actually go into the GO phase, especially your mature muscle and nerve cells. They never divide. Some cells can be called back from the GO phase with some external clues in there, but some cells will never leave that phase in there. So this picture here indicates that if you've got this G1 checkpoint and things go down in there, that allows you, where your gate goes down, you're going to go ahead and continue progressing through. If there's a gate up stopping it, that's when you're going to shift off to this other alternate GO phase in there. So your cell cycle clock is going to use cyclins and cyclin-dependent kinases. So your protein kinases from the last chapter, if you remember, are going to be enzymes that are going to help to activate and inactivate other proteins by phosphorylation. So a kinase is going to be an enzyme that moves phosphates or phosphorylates things. So these may be present at constant concentrations in growing cells, but they'll be in an inactive form. The cyclin is a protein that's going to attach to a kinase to activate it. So your CDKs or cyclin-dependent kinases are going to be kinases that require cyclin, and those will actively rise and fall with concentrations of the cyclins or the concentration of its cyclin partner in the cell. So as your levels of cyclins go up and down, that's going to control the enzymes that are going to control where it is in the cell cycle. So another thing we found is the MPF, or the maturation promoting factor. It can be thought of as an M-phase promoting factor in there. So your cyclin synthesis is going to begin late in the S phase, and it will be synthesized through G2, and then it will be protected from degradation and will be allowed to accumulate. So you can see here you've got those cyclin concentrations increasing in there. The cyclin is going to combine with the recycled CDKs, and then it will produce enough MPF in there for the cells to pass the G2 checkpoint. The MPF is going to promote mitosis by phosphorylating various proteins, and that will peak during metaphase. Then you'll have the cyclin component of the MPF begin to degrade during anaphase, which will terminate the mitosis phase, and the cell will go back into the G1. So you can see here where you've got the cyclin concentrations and the MPF activity increasing for mitosis, and then it comes back down. And then it slowly builds again, the cell goes into mitosis, and then it drops back down. So those are going to help to regulate the cell cycle. So in G1, your cyclin is going to degrade and your CDK gets recycled. And when we look at it in here, you've got your NPF. As you come out of mitosis, your cyclin is going to be degraded in there. As you go along and you come around through the cell cycle, you're going to start to have the cyclin accumulating in there, and it will go ahead and then start your MPF so that you can progress through the next phase of the cycle. So your MPF will act directly as a kinase and indirectly by activating other kinases. So it can cause phosphorylation of various proteins of the nuclear lamina, and that will start to fragment the envelope. It also contributes to chromosome condensation and spindle formation in prophase. So during anaphase, this is when you're getting ready to split into two new cells. The cell is going to initiate a process to destruct its own cyclin in there, and that switches itself off from being in the dividing phase. We know with animal cells they appear to have at least 3 different CDK proteins and several different cyclins at the G1 protein. So some other stop and go signals we have are internal and external signals at the checkpoints. You have internal signals at the M phase for your mitosis phase. During the anaphase separation of the sister chromatids, this does not begin until all of the chromatids are attached at the metaphase plate. So you're not going to even begin to separate them in there. Only when the kinetochores are attached, then the regulatory protein will be activated. The reason for this is when you're separating the chromosomes like that, you don't want to end up with extra chromosomes or missing chromosomes in one of the daughter cells. So that helps to ensure that the two cells are going to both get the equal number of chromosomes and the proper number of chromosomes. So the growth factor is a protein released by certain cells that's going to stimulate other cells to divide. So far, we've discovered over 50 different growth factors. So one example is the platelet-derived growth factor, or PDGF. This is made by the platelets, and it's required for division of the fibroblasts. So when you get an injury, this is going to be released from the site of injury, and the proliferation of the fibroblasts actually helps the wounds to heal in there. So when you get a cut or a wound, your body is actually going to send out the cell to tell those cells, send out the message to tell those cells to divide there. So this is showing the study of it being done with a sample of human connective tissue that's being cut up into small pieces in a petri dish. Enzymes are going to digest the extracellular matrix so that you've got a suspension of these free fibroblasts. Then they get transferred into culture vessels. You're going to have some without the PDGF and some with the PDGF. When you add the PDGF in there, you're going to get growth of the cells. Another thing that's going to regulate cell division is the density in there. With density-dependent inhibition, there's an external signal that's going to tell the cells, it's getting crowded in here, stop dividing. Another signal in there is going to be anchorage dependence. Most animal cells need to be attached to a substrate to divide. They don't like to float around and just divide randomly. So when some of these controls start to go bad, that's when you can have cancer cells produced. One of the things with cancer cells is they do not follow the normal signals regulating the cell cycle. They lack density-dependent inhibition, so that's why tumors will grow in spaces, and they will just keep on growing even though there's not room for them and put pressure on other tissues. They also lack the anchorage dependence. So when you lack anchorage dependence, that allows you to be a cancer cell that floats around and can metastasize or start to grow in another location in there. So a normal cell is going to be done and stop dividing when growth factors are depleted. So if they stop dividing in cancer cells, generally they're going to do so at random points, not at the normal places where a normal cell will divide in there. So normal cells cannot divide indefinitely. However, certain cancer cells can divide indefinitely as long as they have a continual supply of nutrients. So back in 1951, they were looking for cells that they could use in cultures to have grow for a long period of time. And when they would get these cell cultures, most of the time they could divide anywhere from 20 to 50 times, and then the cells would basically stop dividing, they would age, and they would die. There was a woman, Henrietta Lacks, that had a tumor that was collected, and her cells ended up being grown in these cultures. And there is a whole story behind it where she wasn't really aware that her cells were being collected for this. And these tumor cells became something that was used for a lot of research in there because they didn't die. They didn't appear to age in there. They just continued to divide. So it started out with one researcher who was looking for some of these cells that would grow without dying in there. And he just happened to be the one who collected them from Henrietta Lacks in 1951. So he kept these cells alive, and through his contacts with other researchers, he ended up sharing the cells. And he would end up sharing them with other people who would contact them. He wasn't really making money off of them. He was just sharing them in the name of research in there. But eventually, over time, what ended up happening is this strain of cells became more and more widely used, and they were produced commercially in there and being charged for. So it's kind of an interesting story in there. There is a book actually out, what is it called? The Immortal Life of Henrietta Lacks, that's by Rebecca Skloot in there, that talks about how the family ended up finding out about these cells and how they didn't understand what was going on with them. But they're referred to as HeLa cells in there, and these cell cultures like that get part of the person's name in there. So from Henrietta Lacks is where the HeLa comes from. And HeLa cells are still widely used today for research because of their ability to continue to divide in there. They're actually pretty persistent, and a lot of other cell cultures have been contaminated with HeLa cells as well. So transformation is going to be the process that's going to convert a normal cell into a cancer cell. Normally, your immune cells are going to destroy some of your cancer cells in your body. Most of us are going to produce some cancer cells here and there, and our immune system will take care of it. But in some cases, it will evade destruction and proliferate, and then it will form a tumor. A tumor is considered a mass of abnormal cells within otherwise normal tissue. It's considered benign if those abnormal cells remain at the original site. Most benign tumors can be removed by surgery. A malignant tumor is going to be one that becomes invasive enough to impair function at one or more of your organs, and then it's referred to as cancer. So here it's showing the tumor growing in some glandular tissue in a breast in there, and the tumor begins to grow, and then eventually some of those cells will break loose because they're not behaving the way they're supposed to. They're not needing to be anchored to something, and then they will travel in the lymphatic vessels and the blood vessels in there. So because of it traveling in the lymphatic vessels, that's one of the reasons why they will look at the lymph nodes in a cancer patient to get an idea if those cancers are spreading in there. So then it's showing it becoming a metastatic tumor, where this tumor is not it did not originate there, it came from cells that originated somewhere else. So malignant tumor cells are abnormal in many ways beyond just excessive proliferation. They may have an unusual number of chromosomes in there. Its normal metabolism may be disabled, so it doesn't actually serve any useful function. They can change their surface to allow them to lose attachment so that they can spread around easily. They can even secrete signals that will trigger angiogenesis. So they will trigger signals to have blood vessels grow and bring them a supply of food in there. So metastasis is what is considered a separate from the original tumor mass in there, and it will proliferate in a distant location. So how do we treat cancers? Cancer cells are going to replicate faster than normal cells do. So one of the things we try and do is kill these cells before we kill so many normal cells that the person dies. One option is high energy radiation. This can help treat localized tumors because it's going to damage the DNA in the cancer cells more than it will in normal cells. The majority of cancer cells lose their ability to repair the damage in there, and so this will kill some of them. You're going to kill some of the normal cells too, but the hope is that you kill enough of the tumor cells before you do too much damage to the normal cell and lose the patient. Chemotherapy is going to use drugs that are toxic to actively dividing cells through the circulatory system. This is going to be used when there is suspected or known metastasis because it's going to float around more. The radiation can be targeted and localized into just a small area, where the chemotherapy, the idea of having it circulate all over in the bloodstream is that hopefully it will target any loose stray cells that have wandered away in there. And Taxol, this is a drug that freezes the mitotic spindle by preventing the microtubule depolymerization. So this stops the cells from going past metaphase. This can be used as a cancer treatment, and sometimes people are given this after taking chemotherapy in there. It's been used with a lot of breast cancers. Cancer treatments are going to have a lot of side effects due to the drugs having their effect on normal cells. With our methods of treating cancer right now, there really is no way of avoiding side effects in there. Sometimes people will do some other things in there. They'll use some alternative medicine, maybe used in conjunction with traditional cancer therapies, to try to minimize the collateral damage from the cancer treatments itself. But virtually all cancer treatments that we have right now in traditional medicine are going to be quite toxic. So these are a couple of interesting sources that you can use. Nova has a video, it's been out for a while, called Cancer Warrior, and it talks about how they discovered the angiogenesis inhibitors, and it gives a nice explanation of how they work, on why when you remove a tumor surgically, in some cases, the other tumors start to grow in the body where it's been spread in there. And then the book, The Immortal Life of Henrietta Lacks, gives a lot of history on the HeLa cells and how we ended up discovering those and what has happened with them in there. Neither of those are considered testable material in this class, but if the cancer treatment or the research cells are of interest to you, those are a couple of good sources in there that are. The Henrietta Lacks book was actually fairly easy read in there. It wasn't your typical scientific reading in there. And that is the end of chapter 12.